morning. Today we are going to discuss an article from the journal Pain with the title Posttraumatic Hyperalgesia and Associated Peripheral Sensitization After Temporomandibular Injury in Mice. So the title says it's an animal study, and here they follow the arrived guidelines, which has a study design, sample size, inclusion criteria, randomization, blinding, outcome measures, statistical methods, experimental animals, and experimental procedures. After completing these analysis of the journal article, we'll come to analyze this. Right. So the article title, as mentioned, it is a post-traumatic hyperalgesia. Post-traumatic is after an injury, there is a hyperalgesia, increased pain, and there is associated peripheral sensitization, where the nociceptors are sensitized in the peripheral areas. The injury is in the temporomandibular joint, and the injury is happening in the mice. So this study was conducted in the year 2024. Was published in the journal Pain with an impact factor of 7.96. The novelty of this study, so the research gap was the mechanical hyperalgesia was already studied, but there was lack of information regarding the functional outcomes, the neurochemical changes, and the sex differences in the pain responses following a TMJ injury. So this was the novelty, and the aim of this article was to investigate the effects of temporomandibular joint trauma induced by forced mouth opening. So this was a procedure which was followed to create a temporomandibular joint injury, focusing on the pain behaviors and peripheral sensitization while considering the influence of sex on these outcomes. So this comprehensive approach was intended to enhance the understanding of the TMD associated pain mechanisms. So going to the materials and methods, the animal preparation and anesthesia. So they used the C57BL6 mice. So it is a black mice, whereas the spray dolly is a white, which were anesthetized using a combination of ketamine and xylamine, which is 80/10 mg per kilogram for the procedure. A colibri retractor was used to maintain the forced mouth opening for 3 hours each day over a period of 5 consecutive days. And this is the colibri retractor which was used to create the injury. So forced mouth opening procedure was performed by placing again they used this procedure for another type imaging. For that they used this custom made mouth opener, which was like 1.5 centimeters in dimension, and this procedure was repeated either in 5 days or 2 days for comparison. And a sham group underwent the same procedure without the mouth opener. And I think sham group I've already discussed. It is nothing but sham procedure is nothing but a procedure performed on control group participant to ensure that he or she experiences the same incidental effects of the operation or procedure without getting this procedure done. So they undergo all the anesthesia, the isolation, diet, all the procedures are same like the experimental group, but only one difference is they don't undergo the surgical procedure. So this is how they have used the mouth opening injury, forced mouth opening injury was done. And the diet and the monitoring after each FMO procedure, mice were provided with soft diet and water, body weight was monitored closely. And the behavioral assessment for pain behaviors, they assess your Von-Frey test for mechanical pain, mouse Grimace scale for spontaneous pain-like behaviors, and the open field test for anxiety-like behaviors. Von-Frey test, in the last journal discussion, I told about the Von-Frey filament. Here they use this for the mechanical pain analysis, where they can get the pain score here. So this is the clinical picture of doing the Von-Frey analysis for the mechanical pain. And open field for anxiety test. So they have a custom made box, and they have a center region, and they have all the markings here, and they allow the mice and record the movement of the mice using a camera so that they can see the anxiety of these experimental animals. And mouse Grimace scale, which I had already told, these are the scales which are used to detect the pain levels in the animals. And bite forces, this is just a picture to show how they use animals to detect the force of bite. So when an animal bites, it is transmitted to the computer where it is recorded. So when they make the animal bite that thing, it is transferred and it is recorded. So this is the spike was recorded, and calcium imaging was done, which was in vivo, which means the animal is alive, and they do the calcium imaging, which was conducted to observe the changes in the spontaneous calcium responses and mechanical hypersensitivity of the trigeminal ganglia neurons. So this is how they do the calcium imaging. They place the microscope or the fiber optic thing, and they measure the calcium release. So labeling of occurrence. So this they have done to label the occurrence of the trigeminal system, where the wheat germ agglutinin 488 was injected into the TMJ to retrogradely label the trigeminal occurrence. So this labeling was performed before and after the FMO or sham procedures to assess changes in the neuronal proportions and marker expression. So retrogradely labeling means the trigeminal occurrence, it is a technique to trace the pathways of the sensory neurons in the trigeminal nerve from the brainstem back to their cell bodies in the trigeminal ganglia. So from the brain to the ganglia, they are retrogradely tracing the apparent nerves by introducing a tracer substance. Here they have used the wheat germ agglutinin 488. And immunochemistry, histochemistry was performed for after the FMO procedures, which is after sacrificing the animal. For the trigeminal ganglia to analyze the expression of specific markers like TRP1 and CGRP, which is involved. This involved cryosectioning tissues and applying primary and secondary antibodies like ISs. So TRP1 is a transient receptor potential vanilloid. It is a protein that functions as a non-selective cation channel, primarily known for its role in detecting noxious heat and pain, and is activated by stimuli like capsaicin, heat, and protons. Capsaicin is nothing but the chili, the chemical from the chilies. So this is the TRP1 protein, which is seen on the cell membrane, and the structure is there. When the heat or any protons or any noxious stimuli enter here, then there is a cascade of events happening. CGRP is nothing but calcitonin gene-related peptide, which is a neuropeptide involved in various physiological processes, including pain perception, blood vessel dilation, and inflammation, and is a target for migraine treatments. So in the nerve endings, we can see this CGRP is getting released, and they reach the receptors. Here they are the receptor complex. So here they come and they transmit the signals. And this is how the immunohistochemistry is done, along with the calcium imaging. So everything they have done together. So when we analyze the results, they found that there is increased mechanical pain. The FMO procedure significantly increased the mechanical pain sensitivity as assessed by the Von Frey test. So the mice exhibited heightened response to mechanical stimuli, indicating a state of hyperalgesia following TMJ injury. So when the injury is made in the TMJ, later it causes peripheral sensitization and they become more hyperalgesic to the mechanical stimuli. So the results are shown here. We have the FMO group and the sham group, and this is for the 5-day period, and the statistical analysis is shown here. And the spontaneous pain-like behaviors. So the study observed spontaneous pain-like behaviors in mice, which were assessed using the mouse grimness scale, which suggests that the FMO procedure not only induced mechanical hyperalgesia, but also led to behaviors indicating of ongoing pain. So they have the ongoing pain, which is recorded through the grimace scale. And the anxiety-like behaviors, which was noted in the open field test, indicating that the FMO procedure may have psychological effects in addition to physical pain. So this aligns with the observed emotional affective pain changes in the higher order brain centers. So affective pain happens in the central processing, central sensitization. So this is the this open field test which was recorded in the video, and they have shown here. This is the FMO group and this is the sham group. The center part is the area where a mouse comes to rest. So that part is very less in this area. Whereas in the sham group, the center resting is recorded, and the statistical analysis also confirms this. And in calcium imaging, in vivo calcium imaging revealed that FMO increases spontaneous calcium responses and mechanical hypersensitivity in trigeminal ganglia neurons. So this effect was more pronounced in male mice, suggesting a sex-dependent response to TMJ. So here the sex differences was noted. All the other previous procedures, there was no difference between the sexes. So in the calcium imaging, they found the difference. And peripheral sensitization, the results indicated a robust peripheral sensitization of trigeminal nociceptors following FMO, and this was evidenced by increased calcium responses to capsaicin and other stimuli, highlighting the role of peripheral mechanisms in development of pain. So they have compared between the sham and the FMO groups. So the limitations of this study were the sex differences was not fully explored, and there was incomplete resolution of hyperalgesia. So there was hyperalgesia, and when did it come down? That was not discussed in this. And the central sensitization was not assessed in this, and there were model limitations. It may have not fully represent the entire spectrum of human TMJ since it was done in the animal model. And there was a potential bias in retrograde labeling. So when they tried to label the afferent fibers through retrograde method, there was a bias labeling certain subsets of afferents. This could affect the interpretation of the data regarding the contributions of specific nociceptors to hyperalgesia. So some afferents may not be labeled. And there are limited behavioral assessments. They used specific pain-like and anxiety-like behaviors, which may not capture the full range of symptoms. And more comprehensive understanding of the impact of injury needs more behavioral evaluations. The future scope is the study should focus on the neurochemical changes in trigeminal afferents and the functional implications of these changes in both sexes. Understanding these mechanisms could lead to better therapeutic strategies for managing TMJ-related pain. So in conclusion, the FMO was a reliable, they used FMO as a reliable model here. It effectively mimics the clinical manifestation of TMJ arthralgia, including profound long-lasting hyperalgesia and pain-like behaviors. And peripheral sensitization, they have found a highlight that TMJ injury leads to significant peripheral sensitization of trigeminal nociceptors by increased sensitivity to mechanical and thermal stimuli, indicating that peripheral mechanisms play a crucial role in development of pain following TMJ injury. And sex differences in pain responses, they have done the study here. So the strengths were they used relevant animal model, and there were comprehensive assessment of pain behaviors. Investigation of peripheral sensitization was used, and there was a sex-dependent analysis, and behavioral correlation with the clinical symptoms were also conducted. So coming to the guidelines. So here they have discussed about the study design, but they have not mentioned about the sample size in this article. They have just mentioned they used the sham group and the experimental group, but the sample size was not mentioned. They had the inclusion criteria of using 18 to 12 to 18 weeks of mice, both male and female, but the exclusion criteria was not really mentioned. And randomization was done and blinding was also done. They have also mentioned about the outcome measures. The statistical analysis was done perfectly, and then experimental animals regarding the species, they have mentioned the species. Experimental procedures are also mentioned, and the results are clear. So this concludes this discussion.